Hi, this is Jesse Wilson. This is a lecture for Bio120 Microbiology, and this lecture deals with microbial ecology. We are going to go over the different aspects of ecosystems, think about some of the roles that microbes have in these ecosystems. We're going to go over studies that have applied macroecological concepts to microbes, and then finally we're going to discuss why thinking about microbes in an ecological sense is important. First, let's take a step back and think about the different factors that make up ecosystems. The planet is composed of various spheres. The biosphere is all the living organisms on the planet, and there is really only a thin envelope of life on or near the surface of the Earth. There is also a hydrosphere, which is all the water on the planet, a lithosphere, which is all the rocks and soil on the planet, and finally there's the atmosphere, which is all the air on this planet. You can see from this diagram that an ecosystem is composed of many different populations living together, along with the abiotic environment they live in. Ecology is the study of all the interactions of the life forms living in a given area with each other and with the abiotic environment, and so the hydrosphere, lithosphere, and atmosphere come into play here. Many ecologists tend to focus on multicellular organisms, but microorganisms cover every inch of this planet and are interacting with each other, with multicellular organisms, and with the environment as well. Remember that a community is an assemblage of populations that occupy the same space at the same time, allowing them to interact with one another. So there is a microbial community that lives in the soil, pictured here, another microbial community that lives in the open ocean habitat, pictured here, and another community of microorganisms that lives in this pond, pictured here. Remembering that prokaryotes outnumber multicellular eukaryotes by a lot on this planet demonstrates why thinking about them in the environments that we study is important. People are constantly trying to estimate exactly how many prokaryotic cells there are on this planet, and the estimates have varied. In 1998, Whitman estimated it at 4 to 6 times 10 to the 30th cells. This was re-estimated a number of years later, mostly because the number of microbial cells that were estimated to be in seafloor sediments decreased. But this work is ongoing. Keep in mind that this is an estimate of the number of prokaryotic cells on this planet, and not of the number of taxa or species. So of course many of the prokaryotes on this planet exist in soils. This is a table showing you the area that a particular ecosystem covers on this planet, as well as the number of prokaryotic cells that we think exist in that area. You might notice that the ratio of 10 to the 15th cells per unit area is either much below 1 for forest soils, or much more than 2.5 for the other types of soils. This is telling us that the density of prokaryotic cells in forest soils is less than that of the density of prokaryotic cells in other soil types, such as grasslands or swamps. People tend to think of forest soils as having a lot of microorganisms, and they do. They just don't have as many prokaryotic cells as either grasslands or swamps. Fungi, which are eukaryotic heterotrophs, dominate in forest soils. Fungi can deal with the lower quality of carbon that tends to dominate forest soils. This means that there's a lot of carbon relative to other nutrients like nitrogen or phosphorus. And fungi can break down the more complex bonds that are often found in leaf litter. Forest soils also tend to be more acidic than, say, grassland soils, and fungi do slightly better in acidic pHs. However, almost all fungi are aerobic, and so they need well-oxygenated environments, meaning that they won't do well in anoxic environments like swamps or bogs. Bacteria are found much more prevalently in grasslands, tundra, and other soil types. Actinobacteria are a phylum of gram-positive bacteria, and they are very abundant in soils, and so they warrant their own column here, even though they are a type of bacterium. Let's talk a little more about the distribution of prokaryotic taxa or species in various habitats. It should come as no surprise that it is extremely variable. If you go out and look at the microbial diversity in a productive grassland soil, it will likely be much higher than soil that has been subjected to acid mine drainage. But at the end of the day, community diversity is a function of richness and evenness. Richness is the total number of species or taxa that are present, and evenness is how evenly those species are represented in that community. These graphs are showing both richness and evenness. OTU stands for Operational Taxonomic Unit. You can think of this as a microbial species. An OTU is a set of microbes that are at least 97% similar in their 16S rRNA sequence. That's the sequence that codes for their ribosome. Some microbiologists are uncomfortable with the term species for microbes because the biological species concept states that organisms that can mate and have fertile offspring are considered the same species. Because microbes don't reproduce sexually, this creates a bit of a problem when trying to apply this concept to them. But having at least 97% similarity in their 16S means that they are the same operational taxonomic unit. The x-axis here is showing us OTU rank, and it's showing us that there are a little more than 300 OTUs. That means there's a little more than 300 different taxa in this community. This is your taxonomic richness, how many taxa there are. The different OTUs are arranged according to how abundant they are in that community, and the y-axis is showing us the relative abundance of each OTU. Keep in mind that this is a log scale. OTU1 can be found fairly frequently in this community, and as you are parsing through all the sequences, more than 1 out of every 10 sequences will show up as OTU1. OTU2 is the next frequent, and so on. You should see that the microbial community is very rich, but not very even. It has a lot of different taxa, but many of the taxa are not found frequently in the sequence library. That is to say, you might find them once or only a few times in the whole community of microbes. Any OTU that is found less than 0.1% of the time is said to be part of the rare biosphere, meaning that it isn't very frequent in the community. The authors of this paper have listed some different techniques that people use to ascertain OTU richness and evenness. Fingerprinting doesn't actually sequence the base pairs in the 16S. Rather, it just looks at the number of base pairs, or length, of a part of it. Clone libraries are a collection of DNA fragments that have been cloned into vectors and cultured. Today, culture-independent techniques are the most common, and combined with next-generation sequencing technologies, we have been able to delve deeper and deeper into the rare biosphere. Because diversity is a function of both richness and evenness, you can see that on the right, the top graph has a higher diversity, or has a more diverse community, because there are more OTUs present, but also because those OTUs are more evenly distributed. More of them are found more frequently. However, evenness is a lot harder to measure than richness, so many studies that assess microbial diversity will only look at taxonomic richness in a given habitat. Now we are going to go over some trends we have observed in microbial diversity thus far. The first trend is that we tend to see higher species richness in larger habitats. This study assessed how diversity correlated, or how richness correlated, with habitat size, and was really testing whether microbial communities follow a similar pattern to larger organisms, something that's called the species-area relationship. And it's a relatively simple power law. You can see it over here. S is the number of species or taxa in a given location. A is the area that was sampled. C is an empirically derived taxon and location-specific constant. And Z is the slope of the log-log line. All this is saying is that as you increase the area that taxa can inhabit, you will also increase the number of taxa there by some exponential power. For multicellular organisms, Z has been shown to be surprisingly consistent across taxa, but it differs depending on whether you're looking at an island or if you're looking at contiguous habitat. For multicellular organisms, Z is approximately 0.3 for islands, and it's 0.1 for contiguous habitats. The authors here got very creative and found 3 different island habitats for microbes to assess: tree holes, metal cutting fluid sump tanks, and membrane bioreactors. By plotting the island size of the various habitats they tested against the number of taxa, they were able to derive Z values for each habitat type. You can see that they are relatively similar to the Z values that were derived for multicellular organisms that lived in habitats, which is 0.3. For multicellular organisms, we have noted that there's higher taxonomic richness at lower latitudes, near the equator. Think about the biodiversity found in the tropics versus the poles. This could be due to many factors, including that lower latitudes are more stable on a geologic timescale, which could allow for organisms to evolve and not die off from stochastic events as much or as often, and or because it's just warmer there, which allows for faster metabolisms and higher growth rates. But we don't really know. This study here went to many places in the ocean and looked at taxonomic richness to see if a similar relationship exists for microorganisms. While the taxonomic richness tended to be low at high latitudes near the poles, it could be either high or low at these lower latitudes, the equator. So here, sometimes richness was high and sometimes it was low. And so this relationship for microorganisms was really driven by the low richness that we observed at the poles in these oceanic environments. Many similar studies have taken place on land for both prokaryotic and eukaryotic microbes. This particular study looked at fungal richness, and they found that fungal richness was highest at low latitudes. There are a lot of different factors that affect taxonomic diversity, but one of them appears to be the amount of disturbance that a habitat experiences. Disturbances are things that disrupt the status quo of an ecosystem, and can range from mild to catastrophic. An example of a catastrophic disturbance would be something like a fire. Several studies have found that multicellular diversity is highest at intermediate levels of disturbance. That is to say, they are of an intermediate intensity and an intermediate frequency. So these ecosystems are not constantly being disturbed, and the disturbance isn't of a catastrophic nature. This has also been shown for microbes in a laboratory setting. Bottles that were shaken had a higher diversity than bottles that were left unshaken. The reason that diversity is higher at intermediate levels of disturbance is because the disturbance prevents one organism that is better at competing from taking over. Some field studies have also tried to apply this concept to microorganisms, but when you go out in the field with this, you have to cover these really large spatial areas, and so a lot of factors get confounded. As you cover these large spatial areas, there's going to be natural changes in things like soil type and temperature, which can confound your results. Like I said before, the number of taxa in a given area is a function of the number of niches in that place. No two taxa can occupy the same niche in the same space and time. If they try, competition will result, and one will beat out the other. This concept has been demonstrated with multicellular organisms. The most well-known experiment looked at the fundamental versus the realized niches of barnacles in the intertidal zone. You can see that the brown species could potentially live over a wider range than it is actually found. However, the blue barnacle was a superior competitor and so reduced the range that the brown barnacle was actually found in. Competitive exclusion has also been shown in the lab using microorganisms, specifically two types of paramecium. Each species could grow fine individually, but when you tried to grow them in the same petri dish, one outcompeted the other. Competition is only one of many different ecological interactions that organisms can engage in. It is a negative-negative interaction, meaning that neither organism benefits from the interaction. One will eventually win, but it is a battle. For now, be the most familiar with predation, in which one species is harmed and the other benefits, and competition, in which both organisms are negatively impacted. The number of taxa in an area is related to how many niches are in that area. A niche is the ecological role or job that an organism possesses. The niche is a function of the conditions, resources, and interactions that the organism needs to carry it out. So if an organism's job is to use a certain substrate, but that substrate is not available in a certain area, you will not find that microbe in that area. Additionally, if an organism needs to interact with another organism that is not found in a certain area, then it will not be found there. We often speak about niches in a nutritional sense for microbes, and that can be shown a couple of different ways, and each of these graphics does that. You can see very clearly in the left-hand figure that energy flows in one direction, but that's different from elemental cycles. So in any ecosystem, you will have primary producers, the phototrophs, the photoautotrophs, and the chemoautotrophs, and these guys are responsible for synthesizing organic carbon out of CO2. Primary producers, or primary consumers, eat the primary producers, secondary consumers eat the primary consumers, and so forth. If you aren't an autotroph, you are a heterotroph. Decomposers are heterotrophs, but they have the special role of being at the end of the food web, and they take all the organic waste and they turn it back into inorganic nutrients for primary producers to use. And it's in this way that elements cycle through ecosystems, even though energy goes through one way. The picture on the right shows a very common ocean food web. You can see right off the bat that microbes are serving as the primary producers. Dinoflagellates and diatoms are both eukaryotic single-celled photoautotrophs. Because they are single-celled, they are microorganisms, even though they are eukaryotic. Microbes can actually exist in any of the major nutritional niches. There are microbial primary producers, consumers, decomposers, and so on. Microbes are especially important to get decomposition in all habitats on the globe, and they are responsible for almost all of the primary production in the world's oceans. By now you should be familiar with how to classify an organism according to its energy, electron, and carbon source. This way of naming an organism is just getting at their nutritional niche. Photolithoautotrophs, like the cyanobacterium trichodesmium, and chemolithoautotrophs, like Nitrobacter, are primary producers. Fungi are chemoorganoheterotrophs, meaning that they consume organic matter and are extremely important decomposers in terrestrial systems. Finally, many types of bacteria and archaea are also chemoorganoheterotrophs. Actinobacteria is a phylum with many chemoorganoheterotrophic members that is extremely common in soils, but it can also be found in the ocean. All heterotrophic microbes in the ocean play a very important role in marine food webs. In the ocean, there's a lot of dissolved organic matter, DOM, floating around all the time. Sometimes it's called dissolved organic carbon. It is created by three main processes. First, everything leaks. All cells leak. Second, organisms eat very sloppily. And third, viruses lyse open the cells that they infect, primarily bacteria. And when they lyse those cells open, the cell contents are released into the ocean. All of this dissolved organic matter is too small for any of the other trophic levels to use it. The only organisms that can use that really small organic matter are bacteria and archaea. And so these heterotrophic microbes repackage that dissolved organic matter into their bodies, and in doing so, they provide a trophic link so that that organic matter can be reintroduced to food webs. We can describe the growth rate of microbes via the Monod growth model. The Monod growth model shows us how different types of microbes would be affected by different nutrient concentrations or substrate availability. S here is the substrate concentration or nutrient concentration. Ks is the half saturation constant or the nutrient concentration required for that microorganism to achieve half of the growth rate that it could possibly achieve. It reflects the affinity of an organism to take up a substrate and is a function of the surface area to volume ratio. On the graph, you can see that the specific growth rate, or how quickly that microbe is growing, is plotted against the nutrient concentration for two different microorganisms, species A and species B. At higher nutrient concentrations, the microbes are able to grow at a faster rate. Species A has a higher overall growth rate, and it would outcompete B at higher nutrient concentrations, but it grows more slowly at lower nutrient concentrations. These organisms tend to be larger, which is why they are less efficient at lower nutrients. They have a lower surface area to volume ratio. Species B has a smaller Ks, or half saturation constant. This means that it has a higher affinity for nutrients. Species B will grow faster and outcompete A if nutrients are limited. Organisms with smaller half saturation constants also tend to be smaller, meaning that they have higher surface area to volume ratios. Recall that many other factors such as temperature, salinity, and pH also affect cell growth. For temperature, metabolisms and growth will increase with increasing temperature until the temperature exceeds the thermal tolerance of the organism, at which point the growth rate will start to decline. For pH and salinity, each organism has an optimal range. While different microorganisms are capable of different things, it can be quite daunting to measure the individual rate for each organism in a community while also accounting for all the interactions they are involved in. Many times, microbial ecologists will use a black box approach, which deals with how the entire community functions without paying attention to who is there and what each individual is doing. A black box is a system about whose structure nothing is known beyond what can be deduced from its behavior. So you know the inputs and you know the outputs, but that's all you know. This type of model works really well for assessing microbial reaction rates, but you have to be careful about projecting this model to other types of systems or projecting it into the future. So under different types of temperature or pH, you may have a different microbial community that may function differently than what you measured. Something that is related to black box analysis and the microbial loop is bacterial growth efficiency. It is related to the black box model because we estimate bacterial growth efficiency for the entire microbial community, not for one individual species or taxa. Bacterial growth efficiency is just bacterial production over bacterial production plus bacterial respiration. All this is saying is that for the dissolved organic matter, the DOM, that microbes take up, some will be respired so that microbes can maintain cellular structures, while some will be incorporated into their biomass. If the microorganisms are putting quite a bit into their biomass, those microbes are said to be trophic links, and the carbon can be reintroduced to food webs. Conversely, if microbes are respiring a lot, then they're considered to be trophic sinks, so that the carbon that is being taken up is being converted into CO2 rather than biomass. Fungi tend to have the greatest bacterial growth efficiency. Bacterial growth efficiency for very oligotrophic ocean regions, that's nutrient poor regions, is very low and can be around 15% or less. While the bacterial growth efficiency for eutrophic ocean regions, that means nutrient-rich regions, is much higher and can be around 35%. Nutrient-poor regions, meaning a lack of inorganic nitrogen or phosphorus, tend to have low bacterial growth efficiencies because the microorganisms are smaller. They have a high surface area to volume ratio, but they're using a lot of the dissolved organic matter that they take up in order to maintain their cellular system so that they're primed and ready to go for whenever there's an influx of nutrients in the system. They need to be able to take up the nutrients very quickly. So kind of related to whether or not a system is oligotrophic or eutrophic is the quality of the organic matter that is in an area. So low quality organic matter is organic matter that has a lot of extra carbon relative to the nitrogen and phosphorus in that organic matter. So we're going to talk about the Redfield ratio in subsequent lectures, but if there's excess carbon for what a heterotrophic organism needs, then that is considered low quality organic matter. Organic matter that is very low quality, the bacteria are going to typically have low bacterial growth efficiency because they're going to have to be plowing through all of that organic matter just to get at the nitrogen and phosphorus to build their biomass. So that's kind of what this picture is showing here. They have a very high respiration rate relative to what they're producing in terms of their own biomass because they're having to get through all of that organic matter. Thus far, we have gone over diversity and some interactions that microbes engage in ecosystems. But at the end of the day, there are a ton of microbial cells in ecosystems all over the planet, and many of them are aerobic, meaning they use oxygen, and they are chemo-organoheterotrophs, meaning they consume organic matter. This graph is evidence of that. It is showing you atmospheric CO2 concentration over time. You can see that it is increasing pretty steadily because of our use of fossil fuels. But if you zoom in and look at it over the course of 1 year, you see an increase followed by a decrease. If you look very closely, you will notice that the increase occurs during the winter in the northern hemisphere, while the decrease occurs in the summer months. During the winter months, trees shed their leaves, and heterotrophic microbes in the soil decompose the introduced organic matter, using oxygen and producing CO2 in the process. This cycle is stronger in the northern hemisphere because it has more landmass, but a similar cycle is present in the southern hemisphere as well. What I'm saying is that every elemental cycle that exists on this planet has microbial players, and sometimes microbes are the only player for a process, such as with nitrogen fixation. Furthermore, the distribution of microorganisms on this planet and the biotic and abiotic interactions that those microbes are involved in affect these global biogeochemical cycles because they impact the microbes themselves, who are responsible for these rates. We'll learn much more about this in the coming lectures, and as we break down the different cycles, remember to ask yourself who is responsible and what that organism needs to complete that activity. For example, methanogenesis is done by certain types of archaea, and they need a completely anaerobic environment with ample organic matter to do that. This concludes this lecture.